Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Introducing Laboratory Automation into a Regulated Environment from Proof of Principle to High-End Solutions. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Celerion, who developed the content for this presentation. Celerion, a global leader in early clinical research services, offers a unique combination of medical expertise, clinical operation experience, and scientific excellence that gives their clients the confidence to make fast, accurate decisions about their drug development path. For over 50 years, Celerion has leveraged the latest operational concepts and technologies to execute safety, tolerability, pharmacokinetic, and pharmacodynamic studies in highly controlled clinic environments. Their founding mission is to help their clients get their drugs to market quickly so that they touch the lives of their family, friends, and people in need around the world. Now it is my pleasure to introduce you to your speakers for today's event. And first, I'd like to introduce you to Dr. Michael Goschel, Associate Director of Bioanalytical Laboratory with Celerion, Switzerland. Michael joined Celerion in 2012 to support laboratory operations under GXP with special consideration of laboratory automation. Prior to joining Celerion, Michael was the laboratory manager of the Laboratory for Vaccine Research and Development with the Hospital for Children and Adolescents in Nuremberg, Germany. He has been published in more than 60 scientific publications on various topics in bioanalytics. And now it's my pleasure to introduce you to your second speaker, and that is Rebecca Schiebly, Team Leader Automation and ELN with Celerion, Switzerland. Rebecca leads the Automation and ELN team and is responsible for developing and implementing solutions to automate Celerion's diverse bioanalytical assays. She joined Celerion in 2010 to increase user acceptance and active use of Celerion's electronic laboratory notebook. And now it's my pleasure to pass over the mic and the controls to our first speaker. So, Michael, when you are ready, you may begin.
Hello, everybody. Welcome to our webinar about laboratory automation. A recent panel discussion at the Bioanalysis Zone asked about the usage of lab automation in bioanalytical laboratories. The outcome of this talk was that everybody agreed on the benefits, while there's only little distribution of the state-of-the-art lab automation in most companies. Celerion, meanwhile, has a strong focus on using lab automation in its bioanalytical laboratories. In our webinar, Rebecca Schiebly and I want to present our experiences with introducing lab automation and implementing the corresponding standards and processes to our facility in Fair-Altorf, Switzerland. We want to highlight the topics summarized here in the first slide. First, we'll speak shortly about different approaches of lab automation in the industry, with special regard to user requirements and validation approaches. Then we'll present the data flow within our laboratory, the interaction between the robots and the different laboratory database systems. Afterwards, Rebecca will demonstrate our approaches for continuous routine checks and ensure full system integrity. She also will describe our processes of implementation of robotic methods from requesting a new method till its release. I will then focus on very strong achievements of modern automation software, automatic error handling and reporting, and the run scheduling. Finally, we'll present a number of unique applications and summarize the overall automation capabilities of our company. When we talk about lab automation, we need to consider the field of usage. In routine clinical laboratories, the use of fully automated analysis systems has been standard for several decades. The applications are always the same, with little to zero modifications. For this purpose, the laboratory equipment manufacturers have developed excellent systems that are fast, user-friendly, and highly reliable. In the pharmaceutical sector, on the other hand, more open systems are used for laboratory automation in order to apply own bioanalytical methods. These are used then with a similar consistency as before in the routine clinical laboratories. Celerion, as a representative of contract laboratories, has to face permanently and rapidly changing projects, resulting in a high degree of variability for the associated automation. When you ask different people in responsible positions in bioanalysis what they expect most from introducing lab automation, you will certainly hear the same topics. However, the order might be different depending on whom you ask. Increased throughput is always the argument of managers who have to apply for the budget and also the availability of headcount for other tasks, while the robot is performing the assay workup is the most economic argument. For us, in a high salary area, these two arguments are without any doubt extremely important. But also the other achievements in the list, namely assay robustness, data integrity through traceability, and reproducibility are truly valuable. With less repetitions and highest alignment in incurred sample reproducibility. All achievements together result in a high overall reliability of automated processes and robot-created data. We now come to the user requirements, which you should be aware before purchasing the instrument and software. Take your time, request demo sessions of the manufacturer's facility, check software for regulatory requirements, but also for intuitive and user-friendly handling. While the main functionality, liquid handling, is certainly well optimized by the various manufacturers, we saw significant differences in the way how the software of the robot reacts on aspiration errors and how easy this information can be retrieved after the run from the databases for our study documentation. There also might be limitations for integration of peripheral instruments such as centrifuges, washers, other devices, and when they are not from the same brand as the robot, it becomes difficult. So check first about the capabilities and experiences of the vendor when you want to avoid unpleasant surprises. Now shortly about the regulatory guidances. First of all, authorities and customers expect the implementations in compliance with regulations, which is in this case includes both the aspects of computerized system validation, here represented by the FDA guidance on Part 11 compliance, and the AGIT guideline for computerized system validation, combined with all the aspects of bioanalytical method validation as per the EMA guideline. For introducing the robotic system, including all its functionalities into your regulated environment, you must conduct a computerized system validation. Depending whether you expect the system to execute one method only over years, what I call the pharma industry approach, you can add more method-specific tests into the system validation. When you have plenty of methods to develop and to validate, let's call it our CRO approach, you will focus on system-relevant tasks in the system validation, while all method-specific tasks will need to be validated in each method individually. In the graphic, the upper blue quarters are the system-relevant parts, which we executed once and have a general validity for all the following method validations. We recommend to have yet the blueprint method available that can be used to execute all the test requirements for the general system validation, especially file handling, error recognition, data flow, including all the electronic archiving behind, since these ideally should be identical throughout all other and overcoming methods. The lower red quarters display our efforts during bioanalytical method validation on the robot. In brief, we conduct the required standard validation test plus additional tests as shown on the left side, which prove the full validity of the method using the validated robotic systems. A stress test is always included to demonstrate that the machine cannot cause any contamination due to inadequate liquid transfer, and also a maximum capacity test, which demonstrates that the assay can work with a fully loaded labware and reagents without any drift or bias between the first and the last analytical run. Although the methods are mostly based on a master method, yet used for the validation of the system in the upper blue section, method-specific adaptations of liquid glasses or labware may be required and be part of the bioanalytical method validation later. So now it's a pleasure for me to hand over to Rebecca. She will proceed with description of the data flow within our facility and describe the maintenance and control aspects, including the proper documentation in our electronic lab notes. And in addition to that, Rebecca will outline the process of robotic method development from request to final approval of the qualified method.
Thank you, Michael. I want to talk now a bit about data flow and the connection between robots, LIMS, and ELN. And I want to do this with an example. But before I start, I want to share with you the reasons why data flows are relevant. Knowing how information flows between different systems allows Celerion to improve internal procedures as well as to ensure data integrity during the complete automated process. Taking time to elaborate such data flow as the one shown here divides a complex system into its components, and by doing that, it helps you create more efficient procedures aiming to avoid gaps in data integrity. We can only recommend doing such a data flow early on during the validation of your system. Here we show you a Celerion system composed of two robotic liquid handlers, Hamilton and Tekken Evo, our databases, our electronic laboratory notebook, lab notes, and Watson LIMS. In this particular case, the Hamilton system transfers samples from tubes to plates, including performing dilutions, while the Tekken Evo system performs the sample workup, including reagent addition, washing of plates, and plate readout. On one side, we have the clinical samples registered in Watson. And from Watson, a work list is generated, including the sample barcode. This work list is loaded into the Hamilton system. The work list represents one analytical run, and it defines the pipetting order of sample calibrators and controls. To increase throughput, more than one work list can be loaded into our Hamilton system. On the other side, and from our robotic method database, one assay-specific robotic method is loaded that defines the pipetting parameters like volume, mixing, or the source and target position. It basically defines what the robot needs to do. Once the Hamilton system finishes its routine, the Tekken Evo takes over by receiving not only the physical microtiter plates, but also a list of plates IDs. This ensures that the analytical runs are processed in the same order and covers any gaps in the data flow. As with the Hamilton system, an assay-specific robotic method is loaded to the Tekken from our database to initiate the process. Both systems generate electronic output data that are securely saved on our raw data database. On one hand, Hamilton provides a list of pipetting errors that may have occurred while the Tekken generates the raw data from the integrated plate reader. Files are properly identified with the analytical run ID for full traceability. For the evaluation of raw data in Watson, both output files are used, and here we go back to the beginning. Samples with pipetting errors are excluded from the evaluation, and the plate-specific raw data is imported to Watson where the regression is made. The documentation of all the associated processes described here is supported by our electronic laboratory notebook, Lab Notes. I want to focus now on Lab Notes and show you with the following examples how it works. Before, a brief introduction to Lab Notes. Lab Note is an electronic laboratory notebook that is very flexible, and most important for us, it is compliant with Title 21, Part 11 of the Code of Federal Regulation. In Celerion, Lab Notes supports the electronic documentation of various processes via electronic forms, like the one seen here. The specific solutions for each process are developed in-house, making us independent of preset configuration that may not be fit for purpose. In this and in the next slide, I will show you how Lab Notes helps Celerion ensure that equipment routine checks, as well as malfunctions, are properly documented in a timely manner without making the documentation too complex or time-consuming. This electronic form developed in Celerion serves primarily as a guide for the user to document an equipment-related routine task, like the yearly preventive maintenance of our liquid handler. It's seen here as PM. Each section requires specific information that needs to be completed in a sequential order, like the task details followed by the results. Everything that needs to be documented is documented, makes it easy for the user to complete information. A successfully performed task will automatically extend the period in which the equipment can be used. It will also automatically list which task needs to be performed next, in this case, a PECU. Any relevant information can be attached to the electronic form as reference. Here, 5 documents attached. You will see more details about this later. In this example, a malfunction was documented for the same equipment. By documenting this, the equipment is automatically set as out of service in the Lab Note system. This will create a warning if any user tries to document the use of this particular equipment. It will also trigger an email notification to the responsible person, improving the flow of information. Lastly, this is an electronic form for the same equipment, which is used to regularly check its performance. Again, it guides the user throughout the task. The form also ensures that the user documents every material or equipment used to perform the given task. In the next slide, I will show you more details about this particular task, particular form, excuse me. This set of completed forms constitute our equipment logbook, and we can reconstitute at any time which tasks were performed on the equipment. As you can see, all tasks are reviewed by a second user. To improve the workflow, email notifications can be configured within the Lab Note system to request this review. These are some of the features of the Lab Note system that has increased the harmonization of documentation within Celerion, and it has also significantly reduced gaps in the documentation. An additional benefit of the Lab Note system, and a feature that exponentially benefits our documentation process, is the direct connection between Lab Notes and balances. This means that the value from the balance can be directly registered in Lab Notes with one click, avoiding manual entries. Here's a small video for demonstrating how it works. By the way, this is the same electronic form described in the previous slide that we use to document the performance check of the Tekken Evos. Let me put play. So, after all material has been documented, the user selects a balance And then the user records the weight of an empty microtiter plate with one click, and then goes back to the Tekken Evo, where the user will execute a small method to dispense the first volume to be tested. No manual entries. This is the weight. One click. After the liquid is transferred by the Tekken Evo, the microtiter plate is weighed again, and the system automatically calculates the accuracy for this volume. The process is repeated for the second, smaller volume. The liquid used in this case is methyl orange. The pass or fail status for each volume accuracy is calculated automatically, as you can see, following the acceptance criteria described in our standard procedure. To evaluate the precision of the liquid transfer, we measure the optical density of the plate containing both volumes. The evaluation of the OD values is done outside lab notes, but the results are nevertheless registered electronic, in this case, pass. At the end, the user confirms the activity with an electronic signature, including username and password. So I can forward a little bit. The evaluation of the OD values requires a review by a second person. And to facilitate this review, the raw data can be attached to the electronic form as seen here. In this way, the second person has an easy and fast access to the data that needs to be reviewed directly from the form. The process finalizes with the electronic signature of the second user. Again, username and password. So LabNote not only helps Celerion ensuring that our equipment is properly checked on time, but it also is used to document the changes throughout the life cycle of a robotic method, as mentioned before by Michael. By life cycle, we mean the request, testing, release, and retirement of robotic method from our database. The cycle starts with the test facility management approving the creation of a robotic method based, among other parameters, on the number of expected samples. This evaluation allows to properly allocate resources for the development of the robotic method. After this approval, the scientist and the automation specialist compile a list of requirements that the method needs to fulfill, like volumes, incubation times, labware, special consideration regarding stability, etc. Based on these requirements, the method is developed and tested according to our standard procedure. As you can see, everything is a little bit small, but everything is signed. Only after the appropriate test has been successfully executed, the method is released for validation. At any point, new requirements can be added to the existing released method. These are documented and the request is forwarded to the study director. Since the method was already released, this new request is evaluated for possible impacts on running studies. This information is taken into consideration to develop a testing plan. After the evaluation and approval of this testing plan, the changes are performed and tested again. After a successful testing and review, a new version is released and the previous one is archived. This change control process is repeated as required, and LabNotes allowed us to fully trace all changes performed through the life cycle of the robotic method. Now I hand over the presentation to Michael, and I thank you for your attention.
Thanks, Rebecca. So in the next slides, I will focus more on the technical aspects of our robots. First, I want to talk about one feature of the Hamilton Starline robots using the Venus software. The feature is called Total Aspiration and Dispense Monitoring, short TADM, which allows to monitor each liquid transfer step executed by the robot. For this purpose, you have to teach the machine how the pressure curve of the specific liquid behaves at a certain volume during aspiration and dispense. This requires a significant amount of work, as you have to run hundreds of aspiration and dispenses of the liquid, record it, and then draw borderlines as lower and upper limit, which will induce an error recognition once it will be broken through. You can see this in the graphic, how the pressure curves look like for a perfect aspiration, and the green and the red border lines drawn based on these experiences. While running the method under true life conditions, any violation, for example, due to insufficient sample volume, clots, or even completely different liquid conditions, such as severely lipemic samples, will be detected and accordingly reported in an automatically compiled output file. It is noteworthy that this teaching of the machine for different liquid classes is really a challenge and requires very experienced personnel. We have built a huge database of liquid classes to have all standard matrices and all common volumes already covered, that we don't need to do this for each and every new method again. The next important feature is the scheduler of the Tekken Evo. We use this platform to accomplish assays with all integrated peripheral instruments such as washers, incubators, and readers. The scheduler, which is embedded in the software, allows for the scheduling of availability of the required resources on the deck. That means that one plate is on the washer, while another plate gets processed by the liquid handling arm, while another plate might be on the reader. This feature is irreplaceable for us to allow for highest throughput. Apart from this quite common applications such as ELISA plate processing, Celerion Switzerland also has a number of specific robot platforms, which I want to present in the following. First, we have this platform for solid phase extraction, based on a Tekken Evo platform with positive pressure unit integrated. A custom-made programming allows to accomplish the SPE workup for up to 2 SPE plates, utilizing up to 8 different solvents in a method. The platform can handle as well as normal size SPE plates, but also micro-elution plates in a 9612 format, and will deliver the finally eluted analyte in a fresh elution plate, ready for either evaporation or direct injection, depending on the method. So we shortly wait for the movie to end. Now we see how the gripper arm transfers the sample plate in the robot. And to transfer then a sample on the already equilibrated SPE plates. I move a little fast, it goes directly to the SPE, positive pressure unit. Okay. So in the next slide, I want to introduce our unique spiking robot, a liquid handler which can execute the preparation of analyte containing liquids, namely stock and working solutions, calibration standards, and quality control samples. This machine gets programmed based on an SQL database, which contains the source and destination concentrations, the solvents, and of course the volumes, which can easily be up and down scaled on demand. Since the positioning of the associated labware is also defined by the database, the preparation of our standards and controls can be easily prepared with known accuracy and precision of the robots, with minimal bias between different preparations during a bioanalytical study. So until now, we talked about processes and features. Now we want and need to talk about one problem that especially affects us in a CRO. As we drive the robots via Watson LIMS derived work lists, we must rely on readable barcodes on the sample tubes. Differently to handheld scanners, the integrated barcode scanners have not that much tolerance in scanning, since they have one fixed defined position, and barcodes being bent too much to one direction cannot be scanned. On the left side, you see a few typical examples for non-usable barcodes as we receive from central laboratories. Either the barcode is printed without a so-called quiet zone, that means no white space on the borders. So the scanner cannot recognize the beginning or the end of the barcode of the label. Or, unfortunately, but not uncommon, the label is affixed in a 90-degree turn with barcode wrapped around the tube. For these cases, we always have the option to either use a handheld scanner instead or enter the barcode manually when it's not readable by the robot at all. Consequently, we request testers of sample tubes with affixed barcodes before the sample collection starts in the clinic. As we then can give advice, the number of unreadable barcodes dropped significantly. In the middle, you can see how we label our tubes internally, namely for standards and QCs, and below also for plates. The orientation, the orientation is perfectly aligned, and read errors are then consequently eliminated. So shortly, I want to present our automation capabilities at our site. All systems fulfill the Part 11 requirements and are in alignment with GXP regulations. Meanwhile, they have been multiple times inspected by Swissmedic and the FDA, always without any findings. With our approach of worklist-driven and barcode-checked processes, any loading errors can be excluded and a maximum of traceability can be guaranteed. All our lab areas are equipped with designated robotic systems. Even the radioimmunoassays are handled using robots. Majority of analytical instruments, especially for immunoassays, have yet been integrated, including very special ones like alpha LISA readers. So we come to our very next projects. Here we plan to do even more for integration of the remaining reader types via integration of external robotic arms, which will then allow for transferring plates between the liquid handler and the peripheral equipment, which will then not necessarily be within the liquid handler anymore. For our new molecular and cell biology laboratory, we will start with robotic processes to support PCR and fax analysis soon. And finally, based on our profound knowledge, we offer application support for our clients for Hamilton Starline with Venus software. So now we already come to the end. If you are interested to get some more lecture about the described platforms, you can find it and download it from our Celerion website at www.celerion.com/resources. Rebecca and I hope that you enjoyed our presentation, and we are happy to answer any question which you can send us either now or via Celerion Marketing. Thanks for listening.
Well, thank you very much, Michael and Rebecca, for that very detailed presentation. I hope everyone enjoyed it. And just like Michael said, we are now going to begin the Q&A portion of the webinar. So I now would like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of the webinar. I've already received some questions, so I will start with those. So Michael and Rebecca, are you ready? Here is here is our first question. Is there a project you are very proud of retrospectively?
Well, shall I answer?
Yes.
Okay.
Please.
I would say the specific platforms that we shortly described, especially these platforms for creating analyte containing liquids, as our spiking robot, which is absolutely unique in the field. And I think that is something that you really have to seek somewhere else.
Rebecca, did you want to add anything to that?
No, well, every single project has a little bit of a proudness, so I don't have any favorites.
Okay, thank you. Here's our next question from our audience member. The question is there a recurring challenge that is obstructive for general uses of automation?
Yes, definitely to get and to keep the right personnel, because what we described now requires a lot of experience, but also continuous learning, learning from errors and not doing the same errors again, of course. So we are always facing, or we always try to keep the experienced personnel both on the super user or administrator side, which are the programmers and method developers, and also on the lab side, where also experienced lab personnel is required to work consistently and routinely with the machines.
Here I have a little bit to add. It needs to be clear to our audience that there will be always certain automation anxiety when you start introducing these impressive systems. So this is something that everyone has to handle. And there are users that get familiar more quickly than others. And that's the challenge, to identify those persons and keep them, and try to also make the system less scary for everyone, more accessible, more user-friendly. So here in Celerion, we try a lot with trainings, sessions, so that they know what we are talking about, so that they don't see the system as something completely strange, but get familiar with it. So nowadays, now and then, after so many years, we struggle less than before, but yeah, this is a constant challenge.
Okay, thank you very much, Rebecca, for that. Here's our next question. What was the biggest challenge Celerion faced when the first automated process was introduced?
I would say exactly what I just mentioned. There's unknownness. These people are not familiar with the system. They know the bench, they know the pipette, they don't know this big system, how it works. So also, retrospectively, it was really understanding the data flow that I showed you before. Well, the first time you do it, then you start to realize how important it is to have this clear idea. I guess that was kind of the challenge really early on.
Michael, did you want to add anything to that?
Well, I fully agree with Rebecca's what Rebecca outlined.
Okay, all right, perfect. All right, here's our next question. On average, how many bioanalytical assays have you currently automated?
In all laboratories, as we have different lab areas, it's separated in liquid chromatography, mass spectrometry, in all types of immunoassays. This includes radioimmunoassay, but also the plate-based like ELISA, alpha ELISA, electrochemiluminescence. All these together, I would say, best guess is about more than 100, 120. Yeah. Assays, yes, absolutely. Yeah.
Okay, thank you very
And
much.
to be more specific, some of these methods, of course, as we are happy when assays can be reused over time again and again, the same analyte comes back. But there are also some projects where we have only once non-recurring, and then this also adds to this number. But of course, those that show up repeatedly are a major part of our robotic systems.
And just to add, not all of them are fitting the schema show a full automation. Some also are partially automated to take routine tasks away from the analyst. No. So that's why the number I would say, is quite high.
Okay, all right, thank you. Here's another question. What is the biggest benefit Celerion has observed with automation?
I remember last time when we had the FDA in-house, and it was very clear for the FDA inspector that the traceability, so really to read out of the data, as Rebecca has shown, the data flow, but also the data files produced by the machine, what happened at which time, who was logged in, what did the machine do at what step, that makes it so traceable and also very, very reliable to any external person. Of course, for us as a company, also the benefits that were listed on one of the first slides, the higher throughput and the availability of personnel to walk away and do other tasks. These are the things that always come up during these discussions. But I would say we achieved here an extremely high level of traceability, which is especially for us in a regulated environment, for our clients and for our for the authorities, that is by far the highest value.
Well, thank you very much, Michael, for that. Here's our next question. From your perspective, what is the biggest challenge for automation in CROs?
Yeah.
Oh, there's a chuckle there.
Yeah, as we started in the beginning, you see, it's so easy. Sorry to say that, but it's so easy to do that when you work in a laboratory where one machine is doing the same exercise over years, day in, day out. Well, like in a clinical routine laboratory, there they have a sample handler, sample analyzer, which does the same electrolyte analysis hundreds and thousands of times over the year. You won't change it. That's easy to learn. It's easy to handle. We here have projects that appear once, that last, let's say, 2 weeks. Then the samples are measured, the report is finalized, and then we don't see that method anymore, unfortunately, and have already to develop the next one. So the continuous changes, the requests for many different methods in short time, that is what makes it so different for a CRO.
Okay, well, oh, go ahead. Sorry about that.
The development of all what we describe here, you need specialists, and you cannot afford months of programming, testing, development. You required an effective method in weeks, if you're lucky. So it is hard.
Okay, thank you. Here's our next question. For how many years have you been using these systems?
We started to introduce, back in 2013, where we purchased the first Hamiltons. We were first, of course, working with older models from other vendors, which I would not consider as today's state-of-the-art systems. But from 2013 onwards, we developed all these new systems, validated them, brought them online, described and defined our processes to come to the status that we have right now.
Rebecca, did you want to add anything to that last question?
Well, I'm only here for 10 years, so we start with one machine. Now we have more than a dozen
Yeah, I think 14 it's now.
14.
Wow.
So it was an exponential
Increase.
Yeah.
Okay, all right then. Well, thank you very much for those questions. We have reached the end of the question and answer portion of the webinar. If we couldn't attend to your questions, the team at Celerion may follow up with you after this presentation. Now, if you have any further questions, please direct them to the email address that's on your screen, and there it is, marketing@celerion.com. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. And get even more out of today's presentation by downloading the supporting materials available under the Handouts tab. And we have 3 handouts there for you. One is called Introducing Automation to a Regulated Laboratory and Experience Report. The second one is a fully automated workstation for SPE applications, Celerion white paper. And the third handout there is a liquid handling robot for robust and reproducible preparation of standard and quality control. So there's 3 right there for you to pick up, so please go ahead and do so. Now, please join us in thanking our speakers, Dr. Michael Goschel, Associate Director of Bioanalytical Laboratory, and Rebecca Schiebly, Team Leader Automation and ELN, both with Celerion, Switzerland, for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at Xtalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.